But yeah, we're going to talk to you this afternoon about writing, report writing in STEM subjects. So next slide, please, Tanya. So, I mean, I'm here because I have some understanding about where you guys are at in sixth form and where you're going to be moving on to for your undergraduates. So previously I was a science teacher and now lecturer in education and teach teachers. I have a PhD in physical chemistry, so I've got a good, well-rounded background to understand what the needs are, what your needs are at the moment. And also what some of the challenges you might have in writing reports. This is basically just sort of give you a little bit of a heads up about what to expect when you move to your undergraduate courses. Now today I am joined by Sandra, Nena and Afsana. If you would like to introduce yourself, that would be great.
Hi, my name is Afsana and I'm a final biomed student and I specialise in biochemistry.
Perfect. And you're a STEM ambassador with us, aren't you, Afsana,
yes,
as well?
Yes, I'm a STEM ambassador too.
Yeah, brilliant. And then we've also got Sandra.
Thank you, Wendy. Good evening, everyone. I'm Sandra Dias, and I'm a secondary PGC trainee teacher, and I specialize in chemistry. Thank you.
Thank you, Sandra. And Nena?
Hello, my name is Nena Chebu, and I'm the same as Sandra. Although I'm studying the biology pathway, so I'm studying to become a biology teacher.
Brilliant. Thank you so much. So we're going to hear some more from Sandra and Nena, who are going to go into more detail about what a lab report is and how to create a good one. And Afsana is here to give you a little bit of advice on what her expectations and what her experiences have been in her undergraduate degree. So really, writing reports, why do we write them? So once we've carried out an investigation, analysed the data and drawn some conclusions, we need to sort of be able to present all of it to others in a really sort of like comprehensive way. Now, there are conventions that you need to use to when you're writing lab reports for, STEM subjects, and they need to be able to withstand scrutiny from others. They've got to be really clear and concise, use a very sort of specific style of language and a specific style of writing. It's because, you need to make sure that it's understood so that people can peer review it, in essence, so that other people can look at your work who are professionals in that field and experts in that field too, and scrutinize it and make sure that the work that you have done is of quality. So the key things are about being really balanced and really measured. You've got to avoid being sensationalist in how you're presenting. You'll read science reports or STEM reports in STEM subjects, and you'll find that they can often sort of come across as a little bit dry, but that's because we're not writing to create a style. We're writing to report objectively about what we've done. There's got to be careful about what data you use, and you don't cherry pick your best data that supports your thoughts and your own biases, so that you're actually presenting the data in a way which is accurate and representative of what you actually found out through your investigations. And the conclusions that you write as a result of your data must be supported. So Afsana, when you were coming into from your sixth form into your undergraduate degree, had you had much experience in writing reports?
I think the style of reports we were writing were very different to what is seen as a scientific report at university. So during my A-levels, it was very supported, and you were given a set of questions which then you can answer. And so when you put it all together, it's still similar content to what a scientific report is, but you're not creating it yourself from scratch. You're being guided and supported through it. So once I came to university, I had some sort of an idea of what kind of information I should be getting from my investigations and what kind of conclusions I can make and what I can do with that data. But in terms of writing a proper scientific report, I had to learn most of that at university. But it was a good foundation that I got from A-levels.
great. So what did you find your biggest challenge was when it came to writing these reports?
I think it's, again, the style. Like you were saying, it can be quite dry. So when you're writing it yourself, you have to make sure you're not, again, being a sensationalist about it. And you have to kind of be to the point as well, in the style that you write. And also, I think when I was doing it during A levels, there wasn't a great emphasis in writing about the materials that we used, and the methods and explaining all of them, because again, that when we did experiments, that was that section done. We didn't have to write about it. Whereas now at university, I do have to, and doing all the data analysis, it's a lot more in detail, and there's a lot more to write. So I think that took a bit of adjustment.
Okay, and did you find that you were sort of like supported in that process when you joined university?
Definitely. I think as the years went on, it progressed and became more detailed and more difficult. But at the same time, the lectures that you have and the advice the lecturers give you progress in the manner as well. So you get more support as the years go on. And once you finally come to dissertation, which is like the ultimate scientific report, you have extra sessions, extra resources online. We get given checklists. So, we always know what we need to do. And if we have any issues, you can always go to your lecturers to get some advice and help.
Yeah, that's it. It's about having really sort of good structure and systems about how you organise your material, isn't it? Thank you, Afsana. And can we have the next slide, please? So really, like, a well-written report can sort of like really change the way we think about the world and relate to it around us. I've put an example here of Charles Darwin's Origin of the Species, in which he talks about natural selection, and that was revolutionary at the time, and it has filtered through to now we sort of like take it as assumed knowledge, but it's just, there's real power in being able to write properly in the scientific way, so that you can communicate your ideas, and who knows what it is that you're doing may well have legacy in whatever way it might do. Yeah, next slide. So I was just wondering, like, feel free to pop this in the chat, like if anyone, or the Q&A, can anyone think of any other sort of like life-changing science reports that they can think of? And we'll sort of give you 10 seconds to do that. Sorry, Tanya, we can move on to the next slide as well. So we'll do 10 seconds.
Nothing yet, Wendy.
No, that's all right. I'm sure people are thinking about it. So I'm going to pass over now to Sandra and Nena, who are going to talk to you in a lot more detail. Now I've given a sort of a brief overview about what the importance of writing reports. I'm going to let Sandra and Nena tell you more about that structure. So please, over to you.
Thank you, Wendy. So as Wendy and Afsana said, today's session, we are going to cover about what is a lab report, what's the use of writing a lab report, and the different sections of the format of writing a lab report. Next slide, please. So you all are familiar of doing experiments in the A levels as well as in your GCSE. And once you've done an experiment, what do you think you would do next, right? So could you tell me what is a lab report? If any one of you could just roughly say what is a lab report. You've already got an idea by Wendy speaking of it now. So if you can roughly tell me, or can you say in your own words, what is a lab report? You can use a chat function to answer. And I'll be giving you 10 seconds for this.
Nothing yet.
All right, that's no problem. Next slide, please. Thank you. So a lab report is a complete and a detailed record of an experiment. Just as you said, I said you've already done an experiment or you performed an experiment, right? So once you perform the experiment, you need to have a detailed record of whatever you've done. So that's when you put it up or you write it down or you take notes of it. So once you make a detailed record of it, you can share your knowledge or communicate your ideas to others. That's why a lab report is there. So the information what you provide in a lab report should be specific enough, because the audience or the people who read your reports are from various backgrounds. They wouldn't be from your specialized subject, so they would be from different backgrounds. So that's why they would need a hand of the lab report. Next slide, please. Thank you. A lab report also answers the three main basic questions, that is, why you've done the experiment, how you've done the experiment, and what was the purpose or what was the reason for carrying out the particular experiment. And also it also answers the other question of what will you do after finding out the results, right? So what's the main impact of doing the experiment? Next slide, please. Thank you. So as I said, the use of the lab report is as above. The questions as I answered, you'll know why you've done the experiment, what would be the impact. So the impact would be what you've learned from conducting this particular experiment of the day, and why you think the findings would matter. If people would have a look at your lab report, they can replicate and extend the findings. So that's the reason your lab report should be precise and very accurate. Next slide, please. Thank you. Unlike an essay, a lab report has a specialized format, or it's written in a technical way, right? So the lab report for the university students, it's written academically similar to scientists. So scientists have a particular way of writing their papers to publish in journals. So in the same way, even you all at the university level would follow the same format. Next slide, please. Where you have this title, the aim, the abstract, the introduction, the hypothesis, then you'll have the materials and the methods or the procedure, the result, the discussion, conclusion, and the references. Sorry. Yes, so starting with a title. Okay, so what is a title, right? So click, please. Thank you. So a title, it precisely identifies the focus of the experiment of the lab report. So for example, today's session was how to write a lab report. So the title of the session today is writing lab reports in the STEM skills. So that is the focus of today's session. So whenever you write a title, it should present the focus of the experiment or whatever you're doing. Next slide, please. On the screen now, you can see examples of some good titles and bad titles. You have the bad titles on the left and the good titles on the right. Could you just go through these titles and just try to make out what do you think is wrong in the bad titles, or what is missing in the bad titles and is present in the good titles, or why does this make a bad title? If you could just read it out and just put your thoughts in the chat or the Q&A, that would be great. Thank you. I'll give you 10 seconds, maybe 20 seconds for this. You just need to see the difference between, take for example, the first one, just see the difference. Okay, so for example, if you see the first title here, Ecological Studies of Some Northern Lakes, here it doesn't tell you which lakes you should take on, or it doesn't tell you what kind of studies, right? Because ecology is a wide concept. So on the right hand side, it tells you you have to just see about the alga and the eutrophication, and it tells you to go on 3 lakes, right? So the title is very specific. Next slide, please. You should also remember that along with the title, you need to mention your name and the date you conducted the experiment. As you can see, it's presented here in the example of the papers seen there. Next slide, please. The next section of the report is the abstract. Could you click, please? So the abstract, it provides an overview of the report. It's starting right from the introduction up to the conclusion. That is, you have to tell a little bit of everything in brief. Why you do the experiment, what's the purpose, what's the method you've used, what's the result you've got, and what's the conclusion. Since you have a bit of everything, abstract is usually written at the last. Okay, because you have to explain everything briefly in an abstract. Next slide, please. Thank you. So here's an example of an abstract. You can see that it's written everything in brief, right from the introduction to the purpose of the experiment, what they've done, the methods, the results, and conclusion. So if you look at this example, you can say that an abstract has to have around 200 to 300 words. So this summarizes the entire experiment. Next slide, please. Thank you. Nana would be going over this.
So your introduction, what needs to be there? So firstly, your introduction is very important. It sets a good tone for the rest of your lab report. This is where you create a clear and attractive opener. You have to remember, as well as your lecturers and your other people that are well versed in science are going to be reading this lab report, you have to kind of keep in mind that other people that don't understand science as well will need to kind of be carried on, will need to have a little bit of guidance in your lab report. So your introduction is where you start off your lab report journey and you carry everybody along. So I'm going to discuss some key elements of the lab report. The first being your aims. This is where you describe the purpose or the reason as to why you're conducting an experiment. It should be brief and concise and state the purpose without predicting. Reason being is that your hypothesis is where you state your prediction. Your hypothesis is a concept, is an idea that is tested via your research and your experiment. And I'll expand on the hypothesis more later on in the presentation, sorry. Background research. Now, this is a great place to give context to your lab report, to the information that you have discussed throughout your research paper. And you may also decide to include both important and relevant studies in this section. And of course, the significance, the importance of your lab report. Next slide, please. Oh, sorry. Yeah, didn't know there was clicks. So, and as well as your good introductions, you have your bad introduction. So, firstly, not defining technical words, especially words that you are going to use a lot in your lab report. Like I mentioned in the beginning, yes, you have your lecturers and people that are well versed in science reading your lab report, but it's also very important to carry on people that are may not be as well versed in science as well. So it's very good to define key terms. You don't have to define everything. Don't make it into a definition introduction, because you lose your aim and your hypothesis by doing that. Not including your aims or hypotheses. These two are very important. It's basically outlining why you are conducting the lab report or the experiment in the first place. And also missing information and not filling the gaps. Again, with your background, that is a good place for you to fill in the gaps to your readers. Why is it that you're conducting this lab report? Has there been previous studies on it, or even opposing studies on it, just to give your readers a good context of your lab report. Next, please. So to summarize, a good introduction includes clearly defined aim of the experiment, significance of the research is present, the reason why you've done the experiment, background research to provide a broader context to your reader, statement of hypothesis is present. Now, your bad introduction, there are gaps in the studies, significance of research is missing, reasons for selecting the aim is not mentioned, technical words are used but not defined, and statement of your hypothesis is missing. Next slide, please. So, like I said before, I am going to divulge more into hypothesis. So, like I said before, it's a concept or an idea that you are testing throughout your experiment or research. So in other words, it's a prediction. Next slide, please. So there are two types of hypotheses. Your first one being the null hypothesis. Now, this is the one that the researcher tries to disprove. And in fact, researchers try hard not to fulfill the null hypothesis, as the reason being as if they fulfill the null hypothesis, they have to reject the alternative hypothesis, which is what they're trying to prove. So, next, I think you need to click, thank you. So for example, an alternative hypothesis in this case is the application of biofertilizer X increasing the plant growth. The null hypothesis would be the opposite, application of the biofertilizer X not increasing the plant growth. So all in all, null hypothesis assumes that there is no relationship between two variables. And I want to introduce you to three variables that you will see later on in the slides, but I just want to introduce them to you now. So you have your independent, the one that you change, your dependent, the one that changes depending on your independent, and your control, the one you keep the same. Next slide, please. So here are examples of null hypothesis. So the first one being cats show no preference for food based on shape, meaning they'll pretty much eat whatever you put in front of them, irregardless of the shape. Plant growth is not affected by light color, and age has no effect on musical ability. Next slide, please. So if any one of you can you identify which one is the null and which one is the alternative hypothesis? If you would like to put in the Q&A, I'll give you 10 seconds or 5 seconds and move on. Okay, we'll move on. So the light color that has no effect on plant growth is the null hypothesis, and the light color that affects plant growth is the alternative hypothesis. Next slide, please. So to summarize, a hypothesis should be clear and understandable, explain what you expect to happen, be testable and measurable, and have an independent and a dependent variable, as well as a control variable. Next slide, please.
So we're moving on to the materials and methods. So in this section, once you've got the hypothesis, you need to test it. So this is where you will exactly test your hypothesis in the materials and methods. Next slide, please. Could you click 3 times? And one more time. Yes, thank you. We all have eaten cakes and we all love cakes. So if any one of you could just describe or just tell in a few words how to bake a cake in a Q&A function or in the chat function. Just give you 10 seconds for this, and I'd like to move on. Let's wait for 10 seconds. Okay, next slide, please. Yes, the reason for showing you the picture of the cake and asking you to identify, to write down its steps or the method is because we use a precise method for baking the cake. Be it for baking a cake or making a cup of tea, we need the exact measurements, we need the exact quantity. If the ingredients or the quantity goes a bit higher or increases, you don't get the perfect bake, right? Or you don't get the perfect cup of tea. So you need to have the exact amount of whatever you're adding. Next slide, please. So similarly, in science, when you want to conduct any experiment, you have to have a precise measurements and accurate measurements of conducting of making solutions, for example. So here you have a solution of ammonium dichromate being prepared. So when you want to prepare the solution, you will need to explain how you are going to prepare this solution in this methods and materials section. Next slide, please. Along with how you prepare the solutions, you also need to mention what kind of equipments you're using and any kinds of like glassware or any other materials what you're using. Next slide, please. Thank you. You could click it, please. Yes. So this is a section where I've told you as before, you'll describe what you actually did. So how you conducted the experiment once you got the hypothesis. So in science, it's sufficient, it's not enough for you to just carry out the experiment, but you need to even provide in detail how you did it, or if you've done any changes to the procedure, you need to write that as well. So for example, you would be given out a lab handout like this in your labs. Okay. If you see this, it's in active voice. Next slide, please. But when you write down the procedure, okay, of what you've done during the experiment, you should write it down in the passive voice. This is that same thing is highlighted here. So I've highlighted it. It's highlighted in yellow, saying that you should remember that you use the past tense whenever you write the procedure section or when you report something. And when, as I've told you, if you do any changes in your procedure, you should mention it here, because when other people or when the anyone, any other student or anybody else wants to replicate your finding or they want to carry out the experiment again, they would need to have the exact detailed procedure so that they get the exact same results. If you do not try to write what you've tweaked, they wouldn't get the same results. Next slide, please.
So our hypothesis is the hypothesis and the method both linked. So next slide, sorry. So in short, yes, they are. Your hypothesis is part of your scientific method. The hypothesis helps to guide it, in fact. Click, please. Just keep clicking. Yeah, thank you. So for this, in this example, the effect of biofertilizer X on plant growth. So you're in your experimental group, you are actually putting the biofertilizer on your plant. However, in your methods, when it comes down to the control group, you would have written, or it would have said, you're not going to spray it on this plant. Next one, please. In this case, you have the case of the independent variable and the dependent variable. So in this example, you have different amounts of water, that you're going to put in your plant. Now, this part is really important because then it affects your dependent variable, which may affect the size of the plant, the number of the leaves, or whether the leaves die or not. Next slide, please.
So whenever you plan an experiment or conduct an experiment, not all experiments will go as planned. There would be some reasons for that. So we'll look over those reasons now. Next slide, please. Thank you. So the first reason would be a small sample size. Okay, it depends on the sample size. Whether you take up a small sample size, what would happen when you pick up a small sample size for an experiment, it would tend to give a false result, or it would create an error. So wherein you tend to pick up the null hypothesis. So people would need to pick up, accept the null hypothesis, wherein you don't get a chance or you don't get an opportunity to work on the alternative hypothesis. So that's a limitation of having a small sample size. That's why it's always better to have a large sample size when you conduct an experiment. With a small size experiment, you don't get the exact results. The interpreted results of the data wouldn't be precise. Next slide, please. Then second reason would be the correlation and causation confusion between both these terms. Correlation and causation, these both always come together, but they both have different meanings. Correlation means it's the association between two variables, and causation is if there's a change in one, how does it affect, or what's the cause on the other. So if you consider a science experiment, how does a causation affect a science experiment is depending on one variable, whether the other variable would increase if one variable is increased, or the other variable would decrease if if the first variable is decreased, would the second variable decrease or increase. So they have a proportional relationship. If you can look at the example here, the human mind usually has a pattern of creating a regular trend, or just like a train track. So they think that correlation and causation are going together hand in hand, but they don't think there would be something else behind this. So for example, if you look at the slide, they say that cell phones are causing cancer, because in the graph in the center you can see that the amount of cancer cases and the amount of cell phone users increases. So they say the more cell phones you use, the cancers are caused. But we all know that there are other causes or other reasons for cancers being caused, and it's not directly the cell phones. We know that it's sun, or there could be genetic reasons, or there could be reasons of your health and lifestyle. Next slide, please. The other reason would be non-replicable results. As I've told you, when you conduct an experiment and you write the procedure, you should write it in such a way where your results or your experiment can be repeated again, and the results could be obtained in the same way. But there are times when you get non-replicable results, that is, you don't get the exact concise results, or you can't repeat the results. So that's another reason for having a bad experiment. Next slide, please. Thank you. And the last reason would be not having a control group. So if you don't have a control group, what's the thing? You only have an experimental group present. So when you have only an experimental group present and the absence of a control group, you never know if you're doing experiment in the correct way, or you never know if you're getting the results within your experimental boundary, or if the results are going outside the boundary. On saying this, I also want to say that all experiments will have an experimental group, but all experiments will not require a control group. There would be some experiments which will require a control group, and sometimes those experiments would have the control group absent, and that's the time when these experiments get into a bad experiment. Next slide, please. Okay, now let us look into two examples two real-life examples where there are flaws in the scientific method. The first example is about, you must have all heard about this, the faster than light, the neutrino anomaly. And the second one is the link between the MMR vaccine and the autism link. Next slide, please. Okay, so faster than light neutrino anomaly. So you can see that they have, when they conducted this experiment in the 2000s, in the early 2000s, they found that there are particles which move faster than light. We know that light travels quite fast, but when scientists conducted this experiment, they found that there are particles called neutrinos which travel faster than light. So they covered a distance of around 730 kilometers by 60 nanoseconds faster than that would be of light. So when they conducted this experiment, they thought that neutrinos are the fastest particles, not the light particles, photons. But later, when the team, when they went back and checked the experiment again, they got to know that there were two flaws in the equipment. So the fiber optic cable, it was attached improperly, and the clock was ticking quite fast. So they got to know that because of these two flaws in their experiment, this experiment was not a good one. So how did they get to know that? Because they went through the experiment, or they went through the they went to the process of conducting the experiment again. Next slide, please.
So I'm sure that maybe some of your parents may be familiar with this case. This is the MMR, too many M's, the MMR vaccine linked to autism, sorry. So on the 28th of February 1998, Wakefield was a lead author of a study of 12 children with autism that was published in the Lancet. So he basically linked the MMR vaccine to the triggering of autism in children. Can you click, please? And click again. Okay, so in the paper, it did actually say there was no connection between the two. However, Wakefield went on to doing press and basically said that there was a link. And of course, this came with implications. As you can see in the graph, parents around the world were not vaccinating their children because they were very fearful of this new lab report from Wakefield. Next, no, just click, please. Yeah, so there was an outbreak of measles in the UK in 2008 and 2009, and Canada and the USA also suffered an outbreak. I'm not going to ask you to write it here now, but I will ask you guys to look into the COVID-19 vaccines and also look at or identify some articles that may be misleading and some of them that may be factual. Next slide, please. So the reasons why his lab report or his findings were retracted was he was guilty of ethical violations, guilty of deliberate fraud. So again, in his paper, it did mention that there was no correlation whatsoever, but he went on to say that there was falsified facts, science misrepresentation, small sample size. He only studied on 12 children, and data was insufficient. Next, please. So, results. This is where you record your findings and your data, and it's so important that this is clear and concise. Each key measurement needs to be reported appropriately. Now, as you can see on each side, they are key coded with the relevant title on the left-hand side. So, for example, your leading sentence summarizes your results. And then also, we're going to look into this a bit more, referencing your references used to support your results. It's very important that, you know, you try and find references that will support your lab report and your results, because then it will give you more of an understanding as to why you got your results in the first place or why you didn't get your results. And it's also very important that you use the right symbols, like the micrometer. I don't know if you can see the highlighted green, the 414 plus or minus 54 micrometer. But yeah, next slide, please. So there are different ways to show your results. The first is a table. Again, it's very important that you put your measurements, your units of measurements next to the title. And it's very important that you lay it out in a clear and concise way. The next example is your graph setup. So on the x-axis, you always put your independent variable. If that's one thing you remember, it's that. And on your y-axis is your dependent variable. Also, if you have two different lines on one graph, it's very key to put a key or symbol to differentiate between the two. And also label your figures. So as you can see on the graph on your right, below it, it says lift and drag coefficients at various angles of attack. It basically describes your results to readers. So you're carrying them along your results and your findings. Next slide, please. And here's an example of results that we've taken from a lab report. Figures and titles are stated, and it describes what we're basically seeing. So to us, we may not understand or know what we're looking at. So that's why the description and the title below is very important. And also using or providing the magnification value for microscopic images. So yeah, next slide, please.
So we're coming to, so once you've already done your methods, you've done your experiment, you've got the results, so what's the next thing to do? So once you've got the experiment and the results, you need to include the discussion. So this is where you interpret the key results in relation to the aim. So this is in relation to the purpose of your experiment. So you need to have the key results and you need to summarize your findings as well as any limitations. Next slide, please. So what do we need to include in the discussion? And I'll be telling you what needs to be avoided in the discussions. So you definitely have to include the results and their importance. So whatever you found out, the data, whatever you've got, you got to include that and whether it matched your hypothesis, right? So that has to be included here in the discussion. You just need to summarize the main ideas. That is, just need to write in brief about everything, and you don't need to write in detail like how you write the method and the introduction. If you've come across any limitations or you need to acknowledge any shortcomings here in the discussions, because a discussion would be well written when you even include your shortcomings, not only your good things but even your shortcomings. It's when your discussion is considered to be good. And if you have any future directions, for example, you've while doing the experiment or once you've got the hypothesis, if you get any other questions, like if you get any other any other questions which you might want to take the experiment further, you can include them here. Or if you even want to include publish this in a journal, you can include that as your future direction here. Next slide, please. Thank you. So what you need to avoid in a discussion, you need to avoid to reiterate your results. That is, you do not need to state your results again, because you've already got an area or a section where you need to mention your results or tabulate all the data. And once that's there, there's no need to repeat it again. You need to make sure that you don't overinterpret your findings. That's because if you overinterpret your findings, there can be a chance where it goes outside your experiment or beyond the scope of study. And you also should know not to use too much of jargon, because I've already told you that the audience which reads your report would be from outside of your specialization. They would need to have simple terms, but do define the key terms as Nana said, but try to use as simple language as possible. And don't include any new piece of data here. This is not the place to include any new piece of data or new information and add to more confusions. Next slide, please. So once you've added your discussions, you come to the conclusion of your report. This is where you need to say what you've learned from this experiment. So that is a take-home message. So you need to just sum up briefly what you've learned from the experiment. You also need to mention the purpose of the experiment. That is, why was the experiment conducted? So this was the question you started off with the experiment, right? So you start off the experiment with a particular question. You want to research on something. So that is the purpose of the experiment. You need to mention that here. You need to identify the main findings. Identifying the main findings is the answer to this question. So you're in search of something, you found the answer after you're in search of something, you conducted an hypothesis. You did the experiment, you found the answer for that. So that's where you identify the main findings. And you also need to see when you perform the experiment, you go through the procedure, you need to see if your hypothesis is met or not met. So if your hypothesis is met is when your goal is achieved. So that's what you need to include in your conclusion. Next slide, please. We now come on to the reference. So here is where when you write the introduction or anything else in the report, when you write the background research or the background significance, you need to add a citation for everything what you've gone through. Okay, so you need to make sure that whatever you take from the other authors, any quotations or any writings from them, they need to be referenced. Could you click, please? Thank you. As you've heard in the previous session, there are three kinds of, there are different styles of referencing, like the Harvard APA. Your department, as you know, would tell you which kind of referencing they need. Harvard referencing is the one which is, I think, most commonly used. So this is an example of a Harvard reference style. When you would write it in the reference section, you need to mention the author and the year, followed by the title of the book or the journal or the paper, the edition, and then where it's published and the place. When you are writing a statement taken, for example, from this author, so if you write a statement, at the end of the statement in brackets, you'll need to mention his name. So for example, it will be Dudley, followed by the year, and you need to close brackets. Next slide, please. So these are some rules what need to be used for citation. So you need all direct quotations from another author need to be cited, as I've told you. All substantial information taken from another source, this also needs to be cited. The only place where you don't need citation is some well-known facts like Newton's three laws of motion, or even the origin of species, what we've just seen. So they don't need any citation because these are all well-known facts. Next slide, please. Could you click for 3 times? Thank you. This comes to the end of the report writing. Once we've written the report, we also need to keep in mind that we need to check the spelling, grammar, and the punctuation. We need to check that we've referenced everywhere correctly, so to avoid plagiarism. And when there are any calculations included, we need to make sure that we have rounded them to the significant figures. Next slide, please. Thank you. So to conclude today's presentation, I would like to say that we all know that science is a process. So it's a process where we observe things, we hypothesize, we try to experiment it, and then we arrive at the results and we come to a discussion, and we just need to report that. In the similar way it's a similar way how the scientists work. So even when they try to make any concepts, or when they try to build up a theory, they would observe, they would work in the similar fashion like I've explained now. And only when these concepts survive, they would become a theory. If they don't survive, they don't. They just die off, or they just get wiped off. Only the ones which survive, and they would become a theory. Next slide, please. Thank you. So this completes the presentation. If you do have any questions, you can add now.
Thank you, Sandra and Nena. That was really great. While you're putting some questions in, I was just going to ask Asana actually, if you have any top tips for people when it comes to thinking about report writing.
I think, again, planning out your structure. Again, that like there is a set structure, but if you just plan out kind of what you want to include in each section, I think it's always a good idea. And I think also think about how you're going to present your data as well. Because when you have graphs and figures and everything, it's important to make sure you're putting it in the right place. So I was always told you, whichever figure you're going to write about, you write all the text first and have the figure at the bottom. So you should never put your figures and graphs before the text. And think about kind of all your figure legends, because again, that's not something you do in A-levels, or from my experience. So it's good to understand what you should be writing in that. And also I think it's one thing that I do personally is I tend to write everything and do my referencing at the end, because it means I can do my word count. And so when I add the in-text citations, I don't have to then go through trying to figure out what the word count is while taking all that out. And just, I think it's good to have kind of a timeline in terms of what you want to do and when.
I'm just gonna clarify on that though, Asana. Like, you know, you've done all your reading beforehand, and you know where your references are going to go. It's not that you're going to put them all in at the end and do your reading at the end. It's already planned. You just actually add the actual citation in it right at the end. Is that right? Yeah.
What I normally do is I'll write a section, and then let's say I got the data, the information for that section from a certain piece of literature, I'll put the link in just underneath that text. So all the links are in for each website and each article that I've used. And so at the end, I just go back and I know exactly where each piece of text has come from. And it just makes it so much easier to do all the referencing. And in case, you know, you've missed something out, you can always go back to these websites because you've got them all in your document. And I also create a spreadsheet which has all of my links and all my references for each section. So you're just like backing everything up and making sure you have everything.
That's very organised of you, Asana, very organised. Like, I think, you know, if you use something like RefWorks, that makes it really easy because that is online. You can just like really kind of like, you can just pop that in there really easily. So all your kind of literature, not just websites, but anything that you're reading from sort of like scientific papers and journals, you can pop in there because you've got that online resource.
Yeah, I think using RefWorks is a good idea.
It's an excellent idea. RefWorks can know any of those platforms or software and being organised with it. I mean, I love that you've got a spreadsheet as well to go with it as well. Thank you so much, and thank you, Sandra. Thank you, Nana, and back to you, Pius.
Thank you all very much. We haven't actually got any questions, so I guess at this point we would like to end the last session for study skills and STEM. Thank you so much for joining.